Okay, welcome to the recording on circulation/respiration. I believe this is chapter 42 in your textbook. First image here is looking at the comparison between circulatory systems. Now, this is assuming that the organism has a circulatory system. An organism that has a gastrovascular cavity would not have a circulatory system because they don't require it. In these two different types of organisms, we have a open circulatory system on the top of a mollusk. Another organism that would have a circulatory system that was open would be an arthropod. As you can see, the key component of an open circulatory system is that the vessels do not actually complete a circuit. So here and here, the vessels don't complete. And we still have a heart for a pump. We still have liquid moving through it. We still have cells. We still have lymph. But the key component difference is that the blood cells are pumped into tissue mass, and they aren't necessarily contained within the vessels. In a closed circulatory system like the annelid, the blood vessel or the blood cells stay inside the vessels. Lymph will leak out, which we'll talk about once we get to the immune system, but the cells should stay inside the vessels. The advantage of having a closed system is you should have more control of where you're distributing blood within the body and all the nutrients that are inside that blood. An open circulatory system doesn't have that control, so you don't have as many, I would say, distinguished features that help aid in the organism's ability to move through space and time, I guess. Let's keep moving here. The next image is showing a comparison of closed circulatory systems of chordates. The first one is of fish, and in fish you have two chambers. You have one atrium and one ventricle. Atriums, remember, are receiving locations, and ventricles are pumping ones. So on the left-hand side we have the deoxygenated blood, and the right-hand side we have oxygenated blood. And you can see that the pump pushes the blood upward in this image to a capillary bed for the gills, and gills are going to exchange gases for us, so oxygen, CO2. And the reason that it's advantageous that capillaries are the ones that are doing the exchanging is we have an increased surface area. So capillaries are very narrow. We end up seeing about 1 blood cell diameter for a lot of capillaries in the systems of animals. So very tiny, lots of surface area. We're going to see lots of exchange of material, and hopefully it won't be cells. If cells are moving out, we're going to have some issues. And then once it's oxygenated, it returns back to the next capillary bed, and that capillary bed is for the body, meaning it goes to all the different systems of that fish. So maybe it's the brain, maybe it's the eyes, maybe it's the muscles that run the body and the movement of the organism. Regardless, we're going to send all the nutrients where they need to be, including the oxygen. Once it's all used up, I send it back to the heart to get into another pump so that it gets back to the gills in a timely fashion. The next one is the amphibian heart slash reptile, because some reptiles have a 3-chambered heart with a very deviated septum. We'll talk about what a septum is in a second. Atria, again, remember, are receiving, ventricle being pumping. So we see that one large ventricle is going to pump the blood throughout the entire body, whether it's oxygenated locations or deoxygenated locations. In the form of a amphibia, we have the lung capillary beds, just like in reptiles, but we also have the ability for amphibian to breathe through its skin, hence why the skin needs to be moist. So we end up getting this pulmocutaneous breathing system, pulmo meaning lung, cutaneous meaning skin. So we've got the lungs here, and then we've also got skin breathing. We're going to oxygenate that blood in the capillaries, return it back to the heart. Heart's going to pump it back to the rest of the body to get used, and then we're going to return any waste that we need back to the heart again for another pump. In a 4-chambered heart like ours, we have this septum in the center. I don't know if you can see that I picked the wrong color. The septum is going to divide the oxygenated blood from the deoxygenated blood to make it much more efficient. It's super duper important that the hearts of mammals are efficient, and same thing with birds, aves, because you're going to have a homeothermic or a endothermic organism that needs to regulate its body temperature. Hence, it usually has a higher metabolism at any given time compared to a reptile. So we need to definitely make sure that oxygen is available for that high respiration rate, and in turn You end up using a lot of ATP at the same time. Okay, so flow of blood. This image is showing more along the lines of a mammal, of a human even. Keep in mind that any medical image, this would be considered a medical image, will have the diagram laid out from the perspective of the patient. So this picture is showing on the left-hand side labels of right, and on the right-hand side labels of left. Well, the reason that is because the patient is laying on the table in front of you, and that's their right atrium and their right ventricle. So the whole right side of the heart is devoted to deoxygenated blood. Remember, we said atriums are going to receive the blood. So the right atrium is going to receive the deoxygenated blood, pass it over to the ventricle, the right ventricle, and when it does, it passes through a valve that is a doorway. That doorway does a opening that's only going to go one way. So as the blood rushes through, it pushes the door open. If the blood were to rush back, that doorway would snap shut and the blood would not flow through. So we're going to make sure that if it moves into the next location, it can't go back. And if we do have leaking back, a doctor can usually hear that with a stethoscope, and it sounds kind of like a whizzing noise, and that's called a heart murmur when the valves have a defect. It's usually an atrioventricular defect, not necessarily a semilunar, which we'll talk about semilunar valves in a second. So then the ventricle receives that blood, the right ventricle, and then it's going to pump that deoxygenated blood where it needs to go, which is to oxygenation process. And when it does so, it leaves the heart. Any vessel that leaves the heart is an artery. This one's going to the lungs, so it's going to be a pulmonary artery. Goes to the lung capillary beds. We'll talk about the lungs in a little bit here. Oxygenates the blood, returns back to the heart. Well, any vessel that returns back to the heart is a vein. Since it's leaving the lungs, it's going to be the pulmonary veins. Back to the left atrium, receives that blood, goes through the atrioventricular valve into the the left ventricle. Left ventricle has now very oxygenated blood, and it needs to pump the blood. Now, the distance here is much larger because you got to send it all the way up to the head and all the way down to the toes. So this pump is going to have to be much stronger on the left side than the right side. And as you can see in the image, the wall of the left ventricle is much thicker. And you'll see that when you dissect the pigs and you cut it open, you'll be able to see that the left ventricle has much more thickness, muscle, than the right. And in the atrium, there's virtually no muscle because the pump distance is much smaller. So the oxygenated blood can be pumped out of the heart, goes to an artery. This artery is called the aorta. Because of its size, it ends up getting a different name. So this is the aorta. And that aorta distributes the blood to the rest of the body, whether it be up towards the anterior or to the posterior. And then the capillary beds exchange the gases and nutrients that they might need. Returns back to the heart in a vein. And that vein in this system, or in this image, is going to be called the vena cava. And the vena cava is going to distribute that blood back down to, or back up to the heart, or down to the heart, depending on which one you're looking at. I should remind you that the anatomy of an artery and the anatomy of a vein are very different. Veins are going to have valves very similar to this type of image. So we're working against gravity. We need to squeeze the skeletal muscles that squeeze the vein. The blood rushes up. The door is shut when it tries to come back down, and we eventually progress back towards the heart. Arteries, on the other hand, are not going to have valves, and instead they're going to have very elastic walls to them because there's going to be a heck of a lot of pressure in those, especially since the heart is squeezing so hard a very large amount of volume. Okay, so this is the flow of blood. The only valve I have not talked about is the ones that are leaving the heart. So they would be here and here, and those are semilunar valves. Those are the ones that are going to be the ones that prevent backflow into the heart. And when you hear a lub-dub sound of a heartbeat, you're actually hearing the lub, the first noise that you hear when you listen to somebody's heartbeat, is those semilunar valves opening, and the dub sound is when the blood comes crashing back into the semilunar valve. All right, so that's the lub-dub sound that you hear. All right, the next piece is the electrical current of the heart, so how the heart understands how to contract. The first part is the sinoatrial node, which is up here. and it is in the right atrium. That right atrium sinoatrial node is also known as the pacemaker. So if things are goofed up in the heart, one can have a prosthetic pacemaker put in that's run by battery. And when you go to the cadaver lab, you might get the luckiness of seeing a pacemaker. In the last couple of years, we've at least seen 2 patients or 2 cadavers that have had pacemakers. Very interesting to see. And the pacemaker is going to send the signal to the 2 atrium. The 2 atrium then contract pretty much in unison, and they're going to push the blood down into the ventricle. We're going to get a little bit of a lag period where we're going to pass the signal to the atrioventricular node, which is kind of covered up here. Right, so the atrioventricular node, which is between the atrium and ventricle. It's then going to send the signal all the way down to the apex of the heart, which is the bottom here. And then through the Purkinje fibers, which are these things here, the signal passes back upward. So we're going to get a contraction that pushes up. So it's going to be a down push, up push, down push, up push of the heart. And we're going to pump that blood all throughout the body. Okay? And that's why you end up getting, if you see a EKG, which is an electrocardiogram, you end up seeing this sort of thing. What you're seeing is the impulse of the sinoatrial and then the impulse of the atrioventricular, right? There's a little bit of lag in between them to ensure that there's full contraction and a little bit of almost ensuring that the blood gets into the next location that it needs to get into. So for example, in the atrium to the ventricle, we want to make sure that that atrium is completely empty, or at least as empty as it can get. And then the same thing with the ventricle. When it contracts, we want to try to get the maximum amount out. I don't know if you can see it here in this image, but you see that there's all these connective fibers here, and here's the valve, right, between the atrium and ventricle. And you'll see that if you look at some of those external links, when they do the open heart surgery, you'll see some of those connective fibers that are inside the valves holding them open, shut, or in the same spot. And I believe the doctor is trying to repair one of those fibers in a person's defective valve. Pretty interesting. Okay, leads us to respiration systems. And the key for all respiration systems is that they be moist, whether they be in the water or up on land. They have to have water for gas exchange. So you're looking at a conglomeration of arthropods and chordates, some of which live on water and some of which live on land. So the top 2 live in the water. So respiration is very easy, especially with gills. Right? Gills, they feather out. They have high surface area. In the one on the left with the crab or a crayfish or whatever it is that's like that, the gills are interior. Right? So you can see that there's a little pocket where the water is going to come into the organism and it's going to breathe through those gills on the inside. And then in the case of this amphibia, probably a salamander of some sort, we have gas exchange gills on the exterior, but regardless, both of them work very efficiently in the water. If you take those organisms out, those gills compress upon each other and the surface area goes to nothing, and in turn the organism usually suffocates. When you move on to land, you have to have something that needs to have a little bit more structure because you're going to have gravity working against it. So in the case of this arthropod, a praying mantis, you have holes that are in the abdomen of the organism, and those are called spiracles, and they network out throughout the organism's body. They're moist on the inside, and they allow for oxygen exchange to the rest of the cells. And then we have mammals like this rabbit that have lungs that are interior as well, and they branch and branch on the inside. And remember, branching allows for increased surface area, so we're going to have a lot of gas exchange. This is showing how a gill works in a fish. It's called countercurrent gas exchange. So we've got water flow across, and notice that the water crosses the very oxygenated blood first, and then it crosses the deoxygenated blood second. Advantage of this is you're getting continual diffusion of oxygen into the blood. The water is going to have a higher amount of oxygen regardless, so if we're saying that this is 100%, which it's not 100% oxygen in the water, and then the artery might be 90, well then oxygen is going to diffuse into the blood, right, allowing this artery to gain oxygen, but the water then is going to decrease. So maybe the water will decrease down to 70 or something like that. Well, then it crosses the vein, and the vein is deoxygenated, so the vein might only be at 40. Well, now we still get diffusion into the veins, 70 to 40. We're getting the optimal amount of oxygen across those gills. So this is called countercurrent gas exchange. I've seen it multiple times on the AP test, so it's something that you probably should understand as far as numbers go and how the balance works and this system. If you flip it and you end up having the vein first, you're going to get a lot of diffusion right off the get-go, but then you won't get any diffusion when you get over to the artery. Hopefully that makes sense, because I can't see your faces right now. All right, and then you got the human lung system, and you understand it goes trachea down to the bronchus, bronchus being the next two tubes, and then those go into bronchioles, and then we end up down in these little sacs called alveoli, right? So we have an increased surface area. We have a liquidy concoction of mucus, I guess, down there to allow for gas exchange. We have oxygen and CO2 being exchanged down there. On the right lobes, we end up having 3 lobes of the lung on the right-hand side, and on the left-hand side, you actually only have 2. And if you could think about why that is, maybe it'll make sense, but if you look at the right-hand side of the diagram, which is the patient's left-hand side, it's kind of uneven. If you think about what's in the thoracic cavity, there's 2 big organs that are in the thoracic cavity. One is the lungs, and the other is the heart. So in this spot right here, where it looks punched out, that's where the heart would be. So it kind of turns that third lobe into a very little dinky lobe that really doesn't serve a purpose at all. Actually, the only thing that it really does is kind of get infected. There are small amount of branching in there, but very little. If there's defects in the lung, one of which is called pneumonia, you end up getting a bacterial infection. We get excess buildup and breathing becomes harder. Another one is cystic fibrosis, where the mucus of the lungs gets a little too thick and we don't get enough oxygen exchange. The last component then is emphysema, where you end up getting damaging of the tissue and then we don't get oxygen exchange as well. I believe this concludes the entire component of blood system. I don't think there's going to be that many questions on some of the other components that the details are in there, but if you don't understand something, by all means, ask and we'll help you out as best we can.